Welcome to Easy Physiology and Research Pro. Today, we dive into a fascinating yet often overlooked aspect of human physiology, salivary secretion. This intricate process is not just about spit, it's a symphony of molecular signaling, epithelial engineering, and neurohormonal orchestration that keeps your mouth moist, your food digestible, and your health protected. By the end of this video, you'll understand how this fluid shapes your daily life, from taste to diagnostics, and how its disruption can indicate deep systemic trouble. Our journey today follows 4 precise objectives. First, we map out the anatomical and histological complexity of salivary glands. Second, we dissect the 2-stage secretion model. Third, we integrate clinical disorders and their diagnostics. And finally, we explore cutting-edge research in salivary gland biology and its futuristic potential in medicine. Buckle up, this is more than just biology, it's translational science in action. Let's start with where your saliva actually comes from. You have 3 major salivary glands, the parotid, submandibular, and sublingual glands. The parotid glands are located just in front of your ears and mainly produce a watery, enzyme-rich fluid. Just below your jawbone are the submandibular glands, which produce a mix of watery and mucus saliva, and they actually make most of your resting saliva. Then under your tongue lie the sublingual glands, which mainly produce a thicker, mucus-type secretion. Apart from these big 3, you also have hundreds of tiny minor salivary glands scattered throughout your mouth, in your lips, cheeks, tongue, and palate. Even though they're small, they're important for keeping your mouth moist and protected all the time. All these glands are structured like little trees. At the ends are clusters of secretory cells called acini, and these feed into a branching system of ducts that carry saliva into your mouth. This tree-like setup gives a large surface area for secretion, while the ducts act like pipelines to deliver it quickly. It's a smart, efficient design that ensures your mouth is always ready for digestion, speech, and defense. Now that you know where the salivary glands are, let's zoom in and see what they're made of under the microscope. The main working units are specialized cells, each with a unique job. First, there are serous cells. These are shaped like pyramids and packed with enzyme-rich granules. They produce watery saliva that's loaded with digestive enzymes like amylase. Next are the mucus cells, which are more column-like in shape and contain mucin granules. These cells secrete thick, slippery mucus that helps coat and protect your mouth. Then come the myoepithelial cells. These are star-shaped contractile cells that wrap around the secretory units. Think of them like tiny muscles that squeeze the glands to help push the saliva out into the ducts. Finally, we have ductal cells, which line the pathways that carry saliva to your mouth. As saliva passes through, these cells modify it by reabsorbing and secreting ions, kind of like a final filter to adjust its composition. So, the teamwork of these 4 cell types ensures that your saliva is not only produced efficiently but also tailored to suit your body's needs at any moment. Let's now break down how saliva is actually formed. This happens in 2 main stages. The first stage takes place in the acinar cells, which are the cells at the very end of the gland's tree-like structure. These cells actively pump chloride ions into the lumen, the central space, and that pulls in sodium and water along with it. The result is an initial fluid that is isotonic, meaning it has the same salt concentration as your blood plasma. But this is just the raw product. As this fluid flows through the duct system, it enters the second stage. Here, ductal cells get to work. They reabsorb sodium and chloride ions and add in potassium and bicarbonate. But here's the cool part. The ducts are impermeable to water, so the water stays behind, making the saliva hypotonic, less salty than plasma. This final version of saliva is perfect for your mouth. It's watery enough to dissolve food for taste, but not so salty that it dries out your tissues. This 2-stage process is incredibly efficient, and it helps explain why your saliva can change so quickly depending on what your body needs. Now let's explore what's actually inside your saliva. First and foremost, it's mostly water, around 97 to 99.5%. Which helps keep your mouth moist and acts as a solvent. But the remaining tiny percentage is packed with powerful components. You've got electrolytes like sodium, potassium, chloride, bicarbonate, calcium, and phosphate. These help with pH balance and even tooth remineralization. Then come the enzymes, like amylase, which starts breaking down starches right in your mouth, and lingual lipase, which helps with fat digestion. Saliva also contains mucins, which are slippery proteins that lubricate your tongue, cheeks, and food to make swallowing smoother. Your immune system gets help too. Saliva has immunoglobulin A and antimicrobial proteins like lysozyme and lactoferrin that fight off harmful bacteria. You'll even find growth factors that promote healing and help maintain the lining of your mouth. And here's something really fascinating. Your saliva's composition constantly changes. It depends on whether you're resting or chewing, the time of day, and even your hydration status. So, saliva isn't just spit. It's a biologically active, constantly adapting fluid that keeps your mouth and body healthy. Saliva isn't just a passive fluid. It plays a huge role in keeping your mouth and body functioning properly. First off, it helps with digestion. As soon as you take a bite, amylase in your saliva starts breaking down starches, and lingual lipase begins the digestion of fats, right there in your mouth. It also helps shape food into a soft ball called a bolus, making it easier to swallow. Next, saliva is a key protector. It neutralizes acids from both food and bacteria using bicarbonate, which helps prevent tooth decay. Saliva also contains antimicrobial proteins, like lysozyme and lactoferrin, which actively fight off harmful germs. Then there's lubrication. Thanks to mucins, your tongue and cheeks glide smoothly during talking, chewing, and swallowing. Saliva even helps you speak clearly and protects tissues from drying and injury. And don't forget mineralization. Saliva provides calcium and phosphate ions that help repair the enamel on your teeth and maintain a healthy pH. It even forms a thin film on your teeth, called the acquired pellicle, that shields enamel from acid attacks. In short, saliva is your mouth's multitasking superhero. Now let's talk about how your nervous system controls salivary secretion. Your saliva isn't produced randomly. It's tightly regulated by your brain, especially through the autonomic nervous system. There are two main pathways, parasympathetic and sympathetic, and they do very different things. The parasympathetic system is the primary driver. When you taste or chew food, signals travel to your brain and back to the glands through cranial nerves, mainly the facial, cranial nerve 7, and glossopharyngeal, cranial nerve 9, nerves. These nerves release acetylcholine, which stimulates watery, enzyme-rich saliva through calcium-based pathways. It also increases blood flow to the glands so they can keep producing fluid. On the other hand, the sympathetic system kicks in under stress. It releases norepinephrine, which triggers a small amount of thicker, protein-rich saliva using the CAMP pathway. This is why your mouth might feel dry but sticky when you're nervous. There are also reflexes, like when you smell food, chew, or even feel nauseated. That can all trigger salivation. So your brain is constantly monitoring what's happening in and around your mouth and adjusting your saliva accordingly. It's a brilliant system that balances fluid output for digestion, speech, and protection. Now we're diving deeper, right into the molecular level of how acinar cells in your salivary glands actually produce saliva. It all begins when receptors on the surface of these cells get activated. These receptors respond to signals from your nervous system. Muscarinic, M3, receptors respond to acetylcholine, and adrenergic receptors respond to norepinephrine. Once activated, they trigger two different signaling pathways. The M3 receptor stimulates the IP3 and calcium pathway, while the adrenergic receptor activates the CAMP pathway. These signals cause ion channels to open, especially for chloride, sodium, and potassium ions. As these ions move into the acinar cell lumen, they create an osmotic gradient, which draws in water. That's where aquaporin 5 channels come in. They act like water gates, rapidly moving water into the lumen to create the fluid part of saliva. Finally, for protein secretion, the cell uses SNARE proteins. These help secretory vesicles fuse with the cell membrane so that enzymes and other proteins can be released. So in just seconds, the cell responds to a signal, activates its internal machinery, moves ions and water, and ejects its protein-packed payload, all with perfect coordination. So once the acinar cells have produced that initial saliva, it's still not ready to do its job in your mouth. It needs to be refined, and that happens in the duct system. Think of the ducts like a high-tech filter. As the fluid moves through, the ductal cells begin reabsorbing sodium and chloride ions, so those get pulled out of the saliva. At the same time, the ducts secrete potassium and bicarbonate ions into the fluid. Bicarbonate is especially important because it helps neutralize acids and maintain oral pH, protecting your teeth and mucosa. Now here's a key point. The duct cells are not very permeable to water. So even though ions are being moved in and out, water doesn't follow. This is why the final saliva that reaches your mouth is hypotonic, meaning it's more dilute than the fluid in your blood. Interestingly, how much the duct cells can modify the saliva depends on flow rate. When saliva flows slowly, the ducts have more time to adjust it and make it very hypotonic. But if you're chewing a lot and producing saliva quickly, it's modified less and ends up closer to the original acinar fluid. So your body has this built-in speed control system for fine-tuning your saliva, depending on what you're doing. Now let's see how hormones affect your salivary secretion, because yes, your endocrine system also plays a part. The most important hormone here is aldosterone, which helps control the sodium and potassium balance in your saliva. Just like it works in your kidneys, aldosterone tells the ductal cells to reabsorb more sodium and secrete more potassium. So when aldosterone levels are high, your saliva ends up with less sodium and more potassium. Sex hormones, like estrogen and testosterone, also influence the salivary glands. During puberty, pregnancy, or menopause, changes in hormone levels can affect both the amount and the composition of saliva. This might explain why some people experience dry mouth at certain life stages. Then there's your body's circadian rhythm. Saliva doesn't flow the same way all day long. Hormones like melatonin and cortisol help regulate this rhythm. Your saliva flow is usually lowest at night and peaks in the afternoon, which also affects the concentration of proteins and enzymes. Even other hormones like ADH, thyroid hormones, and insulin have minor roles, influencing things like water balance and metabolism in the glands. So overall, your salivary system is fine-tuned not only by nerves but also by a constantly changing hormonal environment. So far, we've seen how nerves and hormones regulate saliva, but did you know that the salivary cells themselves can talk to each other? This is where autocrine and paracrine signaling come in. In autocrine signaling, a cell releases a chemical, like ATP, that binds to receptors on its own surface to fine-tune its own activity. In paracrine signaling, the chemical affects nearby cells instead, creating a local network of communication. One important example is ATP, which is released by both acinar and ductal cells. It binds to special purinergic receptors, helping regulate the amount of fluid secreted, almost like a built-in thermostat. Another key player is prostaglandins, which are lipid molecules produced inside the gland. These influence blood flow and vascular permeability, meaning they control how much water and nutrients reach the cells for saliva production. During inflammation, prostaglandin levels go up, and this can change the composition of saliva or even reduce its flow. Then there are neuropeptides like VIP, vasoactive intestinal peptide, substance P, and CGRP. These are tiny molecules released from nearby nerves that act on the glands to modulate secretion or coordinate with immune responses. So, your salivary glands are not just passive sponges. They're dynamic systems with local control loops that adjust secretion moment by moment, based on what's happening both inside and outside the gland. Now let's look at what powers all this secretory action, blood flow and energy metabolism. Your salivary glands might be small, but they are metabolically intense. Believe it or not, they receive nearly 20% of your cardiac output during active secretion, even though they make up less than 0.5% of your body weight. That's a huge supply of blood, and it's necessary because producing saliva requires a lot of energy and nutrients. When you start chewing or tasting food, parasympathetic nerves not only activate saliva production, but also cause vasodilation, which means the blood vessels widen to deliver more oxygen and glucose to the glands. This is supported by neurotransmitters like acetylcholine, VIP, and nitric oxide. The energy is used mainly to power the NEET-PG pump, which drives ion movement, and remember, ion movement is what pulls water into the saliva. The cells are packed with mitochondria, especially in the ducts, to support this high demand for ATP. During times of peak stimulation, glucose use and oxygen consumption in the gland can increase up to tenfold. But here's the catch. If blood flow is disrupted, the whole system falls apart. In conditions like Sjogren's syndrome or radiation damage, blood supply is reduced, mitochondria fail, and saliva production drops drastically. So, blood flow isn't just supportive, it's essential for the entire salivary process. Now let's understand how reflexes control saliva. You've probably noticed that just seeing or smelling delicious food can make your mouth water. That's a salivary reflex in action. These reflexes are triggered by many kinds of stimuli. Mechanical, like chewing; chemical, like sour or spicy foods; olfactory, smelling food; visual, seeing food; and even emotional or psychological cues like anticipation or anxiety. All these signals travel via sensory nerves, mainly the trigeminal, cranial nerve V, facial, cranial nerve 7, glossopharyngeal, cranial nerve 9, and vagus, cranial nerve 10, nerves, straight to the brainstem, where two special centers sit, the superior and inferior salivatory nuclei. These act like control hubs that process incoming information and send motor signals back out to the salivary glands. From there, efferent, outgoing, signals are sent through parasympathetic and sympathetic nerves to tell the glands exactly what type and how much saliva to produce. The parasympathetic nerves, via cranial nerves 7 and 9, control watery secretion and blood flow, while sympathetic fibers, from spinal levels T1 T3, influence protein content and viscosity. And here's something cool, these reflexes are trainable. Remember Pavlov's dogs? He proved that even a ringing bell, when paired with food repeatedly, could eventually make the dogs salivate on cue. That's called classical conditioning, and your brain can do it too, showing just how adaptable and intelligent your salivary control system really is. Alright, let's shift into how we measure saliva in a clinical or research setting. This is important because saliva flow tells us a lot about oral and even systemic health. The most common method is called sialometry, which simply means measuring the volume of saliva produced over time. There are two main types: unstimulated flow, like when you're just sitting quietly, and stimulated flow, when you're chewing or tasting something. To collect saliva, doctors may ask you to spit into a graduated tube, or they might use tools like Lashley cups or cotton rolls placed at the openings of your salivary ducts. For more precise studies, some clinics use radioactive tracers and imaging. This is called salivary scintigraphy, to track how well the glands are working in real time. Now, what counts as normal? For unstimulated saliva, flow should be about 0.3 to 0.4 milliliters per minute, and for stimulated flow, it should be 1.5 to 2.0 milliliters per minute. If unstimulated flow drops below 0.1 milliliters per minute, it's considered hyposalivation, a hallmark of conditions like Sjogren's syndrome or drug-induced dry mouth. Interestingly, saliva flow also varies with the time of day. It's lowest during sleep and peaks in the afternoon. So if someone complains of dry mouth, doctors will assess both objective measurements and subjective symptoms to get a complete picture. After all, what you feel and what your glands produce don't always match up. Now let's look at what happens when salivary function goes wrong. There are several important clinical disorders linked to saliva. One major condition is Sjogren's syndrome, an autoimmune disease where the body's own immune cells attack and destroy the salivary glands. Over time, this leads to severe dryness of the mouth, difficulty swallowing, frequent infections, and even tooth decay. Another common issue is sialolithiasis, which is the formation of salivary stones, basically mineral buildups that block the duct, especially in the submandibular gland. These can cause painful swelling, especially while eating, because the saliva has nowhere to go. Then there's sialorrhea, or excessive salivation. Sometimes it's not overproduction but rather the inability to control saliva, which can happen in neurological conditions like Parkinson's disease, ALS, or cerebral palsy. For some people, this can be embarrassing or cause skin irritation around the mouth. And perhaps the most serious of all is radiation-induced xerostomia, which affects almost every patient receiving radiation therapy for head and neck cancers. The radiation often damages the serous acinar cells, leading to permanent dryness. This greatly impacts oral health, nutrition, and quality of life. These disorders remind us that saliva is not just a comfort, it's a critical fluid for health. When it's missing or disrupted, the consequences go far beyond a dry mouth. Now let's talk about how medications can affect your salivary glands, because many drugs, even common ones, can reduce saliva flow. In fact, over 600 different medications are known to cause xerostomia, or dry mouth. This is especially common in the elderly, since they often take multiple medications, a situation known as polypharmacy. The most common culprits are anticholinergic drugs, which block the action of acetylcholine on the salivary glands. This includes medications like atropine, hyoscine, and even some bladder relaxants. Then there are antidepressants and antihistamines, which can also have anticholinergic effects, drying out the mouth as a side effect. The good news is that in 15 to 30% of cases, simply reducing the drug dose can improve symptoms without compromising treatment. On the flip side, we also have drugs called sialagogues. These are medications like pilocarpine and cevimeline that stimulate saliva production by activating muscarinic receptors. So, understanding the drug history of a patient is essential when evaluating dry mouth. Sometimes it's not a disease, it's the medication. That's why careful review and targeted therapy can make a huge difference in quality of life for someone dealing with salivary dysfunction. Saliva isn't just for digestion and oral hygiene anymore, it's becoming a powerful tool in medicine. Since it's easy to collect, non-invasive, and contains molecules from across your body, saliva is now being used in diagnostics, especially in point-of-care testing. For example, during the COVID-19 pandemic, saliva-based PCR tests became widely used to detect SARS-CoV-2, and similar tests can also detect HIV, hepatitis, and other infections. But that's just the beginning. Scientists are now identifying salivary biomarkers, proteins, RNA, and metabolites that reflect systemic conditions like diabetes, autoimmune diseases, and even cancers. Saliva can help screen for early signs of oral cancer or pancreatic cancer, providing crucial clues before symptoms appear. On the therapeutic side, salivary glands are being explored as delivery systems for gene therapy. Since they're accessible and enclosed, they can be targeted to produce and secrete therapeutic proteins, either into the mouth for local treatment or into the bloodstream for systemic conditions. So, from diagnostics to therapeutics, saliva is being transformed into a clinical powerhouse. In the near future, a simple spit sample may help monitor your health, detect diseases early, or even deliver life-saving treatments. All from your own salivary glands. Now let's take a look at the cutting edge of research, where science is transforming how we understand and use saliva. One of the biggest breakthroughs is in salivary omics. That means studying all the proteins, genes, metabolites, and microbes in your saliva. Researchers have now identified over 3,000 proteins in human saliva, many of which may become future biomarkers for disease diagnosis or monitoring. Another major discovery involves aquaporins, especially AQP5, which are water channels in the membranes of acinar cells. These channels are critical for water transport, and if they're defective, due to genetic mutations or inflammation, it can severely reduce saliva flow. This opens new doors for treating dry mouth by targeting aquaporins. Meanwhile, bioengineers are building artificial salivary glands using 3D scaffolds, stem cells, and organoid technology. These lab-grown mini-glands are starting to mimic real salivary tissue in structure and function, and could eventually be used to restore saliva production in patients with irreversible gland damage. Another exciting frontier is single-cell RNA sequencing, which has revealed previously unknown cell types and developmental patterns within the salivary gland. This high-resolution view helps us understand how glands develop, how diseases progress, and how we might one day regenerate damaged tissue. So, while saliva used to be overlooked, it's now becoming a gateway to precision medicine, tissue engineering, and molecular diagnostics, and the science is only just beginning. Let's wrap up everything we've learned about the physiology of salivary secretion. First, we saw that salivary glands, both major and minor, are structurally and functionally designed for efficient fluid production. From the specialized acinar cells that initiate secretion to the ductal cells that fine-tune it, every part of the system is highly coordinated. We explored the two-stage secretion model, how saliva starts as an isotonic fluid in the acini and becomes hypotonic after ductal modification. Then, we saw how this fluid is much more than water. It carries enzymes, immune factors, minerals, and mucins that support digestion, protect tissues, and even help with speaking and swallowing. Control is multi-layered. Neural pathways, hormonal influences, and local signaling all adjust salivary flow in real time. We also learned how blood flow and metabolism are tightly coupled to secretion. When either is disrupted, salivary function can collapse. Clinical disorders like Sjogren's syndrome, sialolithiasis, and drug-induced xerostomia highlight just how vital this fluid is to our daily health. And finally, we saw how saliva is emerging as a frontline tool in diagnostics, gene therapy, and regenerative medicine. With advances in molecular biology and bioengineering, saliva is no longer a passive secretion. It's becoming a powerful platform for future healthcare. So next time you swallow or taste your food, remember, behind that simple act is a beautifully complex physiological masterpiece. And keep learning with Easy Physiology and ResearchPro.